you, and welcome back to the second lecture of biosignaling. We'll be discussing signal transduction in this lecture, and the contents of this lecture are cellular communication and the definition, phases, properties, and events which are particular to the signal transduction pathways, and the mechanism of action of group 1 and group 2 hormones, how they do their function within the human body. Before we discuss the signal transduction, its various aspects, we need to know how cells communicate with each other. When the cells will communicate, they do communicate with the help of the messenger molecules which they secrete, and this messenger molecules goes from the cell which secretes it to the cell which is the target, and this enables the communication apart from other communication which is direct one. So indirectly, the messenger molecules, which here are called as hormones, as we have already seen in the first lecture of this biosignaling lecture series. The hormones are secreted by the cells, and depending upon whether the target cell is located close proximity or it is far away from the secreting cell, this extracellular messenger molecule has to either travel shorter distance and go to the cells which is in close proximity to do its function, or it has to travel and traverse throughout the body and potentially then go into the target cell, stimulating it and doing its function. What encompasses signal transduction is actually the information superhighways located within the cell. It is able to communicate with different organelles within the cell in response to the hormone which just bound to it, to the receptor, which is either located outside the cell or within its cytoplasm, as we have already seen in class 1 and class 2 hormones. So actually it involves the transmission of this molecular signaling from cellular exterior to its interior and then communicating within the cell's internal domain. It is because of the interaction of the receptor with ligands first, and then the receptors activated, which is then transferred to the different kinds of proteins located inside the cell. Each protein in the pathway will typically act by altering the conformation of the subsequent downstream proteins. So there is a run of proteins which is involved in this signal transduction in this information superhighway, and usually the alterations which happen in the proteins because of the activation either enables it to function at a higher rate or inhibits it. So the signal transduction functions by responding to the receptors which are activated by the ligands. Here ligands are hormones. In general, signal transduction has three phases depending upon what type of a hormone receptor it will be, whether it will be outside or inside. Here the example is for class 2 receptors, and most of the signal transduction is belonging to class 2 hormones, where the receptors are located outside the cell membrane, facing outwards, so that the hormones will bind to the receptor on the outer surface. And it has three phases. First is the reception. Reception involves the binding of the ligand or hormone to a receptor, forming receptor hormone complex, then activating it. And then there is a transduction, which involves the activation of a number of different kinds of proteins within the cell. This whole activation and relay of the information to the final response is called as transduction. This is the actual transduction part. And then the third phase of signal transduction is the activation of a particular cellular response in response to the hormone which is being bound to this target cell. So the relay of the molecules will be more than 3 or 4 proteins which will be activated in response to the hormone receptor complex, and this encompasses the signal transduction in its true sense. The signal transduction which takes place within the cell has 4 distinct properties which enable it to have a tight regulation within the cellular communication. First and foremost is the specificity, that each ligand which binds to the receptor elicits a targeted response, which means that a specific response or specific kind of a metabolism or specific kind of an expression of the gene is specific to the hormone which binds to the receptor outside the cell, and it elicits a specific proteins located within the cellular cytoplasm. Second is the amplification cascade. It means that a single molecule, after activating, activates multiple kinds of molecules downstream to it. So in each step of the cascade, there is an amplification of the molecules which get activated or deactivated. Third is the desensitization, which means that after the action is done, the receptor or the molecule which is within the signal transduction cascade itself gets deactivated, so that there is no further response. This is important in the regulation of the hormone responses. Then fourth one, which is important in the cascading mechanism, is that there are multiple molecules which can do the same function. So they sort of integrate their activation so as to elicit same kind of or similar kind of an or only one kind of an response. So these are the four important properties which a signal transduction has. How does signal transduction work in a cell? We know that it involves the series of events and components which are located within the cell, and they get the transmission to be done from the receptor which is located outside the cell. After the membrane or cytosolic receptor which is there in the cell gets binded to the specific hormone or a ligand, it gets activated, forms hormone receptor complex. This complex in turn activates the signal initiator which is bound to it in most of the cases. After signal initiator is activated, the initiator in itself then activates a number of different kinds of mediators or effectors, which in turn then go and target this specific molecule for its expression or for its release or for its subsequent change in the conformation, and in turn all these lead to the action against a ligand or a hormone which is being secreted on the surface of this target cell. So in turn the action is either the contractility or the release of certain kinds of so many kinds of mediators, or usually the expression of the some genes which affect the metabolic aspects of the cell. As we have already covered in the first lecture of the biosignaling, that hormones can be classified into two different groups, group 1 and group 2, depending upon the receptor's location on the surface or on the cytosolic of the cell. If the receptor for a hormone is intracellular, usually they are the lipophilic ones and they belong to group 1, which are steroids, and they form the hormone receptor complex well within the cell. And for the group 2, the receptors are located in the plasma membrane usually, and they get activated on the plasma membrane, and in turn the effector and the initiator molecules are secreted or activated, which are called as second messengers. And most of the signaling transduction is actually involving these kinds of hormones, the polypeptide or protein or catecholamine hormones, which cannot traverse the biomembrane but act on the surface of the plasma membrane at certain specific receptors and elicit a specific response to them. For the group 1 hormones, the mechanism of action is based upon the receptors which are located well within the confines of the cell membrane. As we already know that group 1 hormones are lipophilic in nature, they are carried through the bloodstream by specific carrier proteins which are located in the plasma. They enter the cell by diffusion through cell membrane because of their lipophilic nature, and when they reach inside the cell they interact with specific receptors which are specific to the hormones secreted, and after interaction they form the hormone receptor complex, which is the one which does the function for them. So hormone receptor complex is the final one which does the action in the target cell, and usually their duration of action ranges from hours to days. Talking about the receptor for these group 1 hormones, how the mechanism of action encompasses within the cell is that these receptors which to which these group 1 hormones, specifically steroid hormones, bind, are actually functioning as the transcription factors. So receptors have two separate domains. One domain is for the interaction with the hormone in itself to form a hormone receptor complex, and after binding to the hormone, it forms a hormone receptor complex, undergoes conformational change to make the second domain available for the interaction to the DNA. As I told you that they are functioning as transcriptional factors, so their main function is to activate the transcription of certain genes which are under the influence of these steroid hormones. So in absence of the hormones, the receptors will not be able to bind to a specific DNA sequence, and they will not be able to do their transcription activation. So after only the hormone will bind to the receptor, forms the hormone receptor complex, the domain which binds to the DNA is exposed, and then it can do the transcriptional factor function. As you can see in this picture, what I just told you, that the hormone will bind to a specific receptor after traversing it to the into the cytoplasm of the cell, forms the hormone receptor complex which will move into the specific area of a DNA, and this receptor will function as a transcriptional activator, causes the transcription of certain kinds of genes, leading to more and more mRNA, and hence consequently the protein or the enzyme which functions under the influence of these steroid hormones. This is one of the mechanism of action and signal transduction for lipophilic hormones. For the group 2 hormones, the mechanism of action is intricate, and this is why the signal transduction is actually visible in their mode of action. As we already know, they are hydrophilic in nature, they cannot traverse the membrane, so they act on the receptors which are located on the surface of the cell by forming a hormone receptor complex on the surface of the cell. Then they are capable of eliciting the varied responses which are dependent on the type of a receptor on which these hydrophilic hormones will interact. Since these hormones are faster to act, they have a shorter duration which ranges from seconds to hours only, and afterwards the action is ceasing to happen, and we need more and more hormone to be synthesized or secreted and to bind to the receptors for continuing their action in the target cell. The receptors particularly are the ones which are expressed on the surface of the target cell, and the hormone interacts on the surface, and they usually are the integral membrane proteins, and these integral membrane proteins are specific to a specific kind of a class 2 or group 2 hormones, so that the varied functions can be happening after the hormone binds to these specific receptors. And each receptor has a downstream cascade of different kinds of proteins which are specific to its activation, as well as the second messengers which relay the information from the surface of the cell to the internal intricacies within the cell. So the binding of the hormone and the receptor is also reversible. After the action is done, the hormone can be degraded and the receptor can be reused for the further cascading mechanism. As you can see in the slide on the mode of action of the group 2 hormone, you will see in the first step the protein hormone will bind to the receptor on the external surface of the cell. It cannot in itself traverse the membrane because of its hydrophobic nature. So the receptors, which are the transmembrane proteins, are located there for its interaction. It will bind to the receptor which is specific to this hormone, form the hormone receptor complex, and by doing so it activates this receptor. The activation is then relayed to the initiator molecule, which many hormones have the associated with the GPCR protein, and here the initiator protein will get activated with the help of an ATP. This in turn will synthesize the secondary messengers in the cascade mechanism, which in turn will activate a number of kinds of different kinds of proteins, usually the kinases, which do the action for the hormone. So the hormone action is relayed through initiator and through second messengers in group 2 hormones. This whole mechanism of activating a number of different kinds of proteins and second messengers is actually the signal transduction or signal cascading mechanism, which brings out the production of the action in response to the hormone for which this target cell is responding. So this was the essential aspects of the signal transduction and the mechanisms of two different classes of hormones. You can visit my Amazon page for the various books on biochemistry and molecular biology, which might be beneficial for your studies. And do subscribe to my channel for further videos which I will upload in due time.